Modern humans like to imagine our origins as a clean line, one species evolving neatly into another, a branching tree with tidy labels. But ancient DNA has shattered that illusion. The deeper scientists look into our genetic past, the stranger the story becomes. Because the first human may not have been fully human at all. For decades, paleoanthropologists believed that species like Homo erectus, Neanderthals, and eventually Homo sapiens followed one another in a slow, orderly sequence. The fossil record seemed to support that narrative. Bones were compared, skulls measured, jawlines debated. Each discovery was placed somewhere along an evolutionary ladder. But bones can only tell part of the story. In the early 21st century, a technological revolution quietly began rewriting human history. Instead of relying solely on fossil shape, scientists began extracting fragments of DNA from ancient bones, genetic material tens of thousands, even hundreds of thousands of years old. And what that DNA revealed was not a ladder, it was a web. The first shock came when researchers sequenced the Neanderthal genome. Living humans outside Africa were found to carry Neanderthal DNA. Interbreeding had occurred. The species were not entirely separate. That alone was revolutionary. Then came something even stranger. In 2010, inside a remote Siberian cave known as Denisova Cave, researchers discovered a tiny fragment of bone. It looked unremarkable. No complete skeleton, no dramatic skull, just a finger bone fragment buried among layers of sediment. But when scientists extracted DNA from that bone, they found something astonishing. The genetic sequence did not match modern humans. It did not match Neanderthals. It belonged to something else entirely, a previously unknown human lineage. They called them the Denisovans. At first, the discovery seemed like just another branch of the human family tree, a sister group to Neanderthals, a population that split from a common ancestor roughly 400,000 to 500,000 years ago. But as more genetic data poured in, the timeline grew murkier, because hidden within Denisovan DNA were fragments that did not match any known human species. Segments far older than Neanderthals, genetic signatures that suggested something unexpected. The Denisovans themselves had ancestors who were not fully like them. Deep inside their genome was evidence of an even older interbreeding event, a ghost population, a mysterious hominin that diverged from the human line nearly 1 million years ago, long before Neanderthals emerged. This was not just interbreeding between cousins. This was something far more ancient. Using advanced computational models, geneticists began comparing Denisovan DNA to modern humans, Neanderthals, and other ancient sequences. What they found was a pattern of genetic segments that were too divergent to belong to any known species. These fragments had been isolated for hundreds of thousands of years before mixing back into the Denisovan lineage. In other words, Denisovans carried DNA from a species that split off from the human line perhaps 900,000 to 1 million years ago. Who were they? The fossil record from that era is sparse. Around 1 million years ago, several archaic humans roamed Africa and Eurasia. Homo heidelbergensis is often considered a common ancestor of Neanderthals and modern humans. But some fossils from that period show anatomical features so unusual that classification becomes controversial. Certain Asian fossils, massive jaws, thick skull vaults, don't fit neatly into known categories. For decades, they were labeled variations of Homo erectus or early heidelbergensis. But what if some of those fossils represent the mysterious ghost population detected in Denisovan DNA? If so, this would mean something profound. The first humans to mix with our ancestors were not just slightly different, they were deeply divergent, closer in time to early Homo than to us. The discovery forced scientists to reconsider a fundamental assumption, that human evolution moved in isolated branches. Instead, ancient Eurasia may have been populated by multiple human species overlapping in time, occasionally meeting, sometimes interbreeding. And one of those species, older than Neanderthals, older than Denisovans, left its mark not in bones we can confidently name, but in fragments of DNA that survived in another species. A species that was not fully human as we understand it, but not entirely something else either. The deeper researchers analyzed the Denisovan genome, the clearer the signal became. Roughly 1% of Denisovan DNA appeared to originate from this unknown archaic hominin. That may sound small, but in evolutionary terms, it represents direct contact, reproduction, viable offspring, and that raises an unsettling possibility. If Denisovans could interbreed with this deeply divergent population, and modern humans could later interbreed with Denisovans, then part of our genetic heritage may trace back to a species that split from our lineage nearly 1 million years ago. Not a side branch, but a shadow lineage, absorbed into us through layers of ancient hybridization. The first human who wasn't fully human may not have been a single individual. It may have been a hybrid, a bridge between worlds that modern science is only just beginning to uncover. And the evidence does not end in Siberia. It spreads across continents. But to understand how deep this hybrid story truly runs, we have to look beyond Denisova Cave and into the DNA of people living today. The most surprising twist in this story does not come from ancient bones. It comes from us. When scientists expanded their analysis beyond Denisovan remains and began scanning the genomes of modern populations, especially in Southeast Asia and Oceania, a startling pattern emerged. Indigenous groups in Papua New Guinea and parts of Melanesia carry up to 5% Denisovan DNA, far more than most other human populations. But buried inside those Denisovan fragments were even older signals. Geneticists noticed certain DNA sequences that were too divergent to fit the Neanderthal-Denisovan modern human split. These sequences appeared to trace back to a lineage that separated from our ancestors roughly 900,000 to 1 million years ago. To put that into perspective, modern humans and Neanderthals diverged only around 600,000 to 700,000 years ago. This mysterious population was older than both. It was as if Denisovans had inherited genetic material from a species that had been evolving independently for hundreds of thousands of years before ever meeting them. Researchers call this phenomenon superarchaic introgression. It is a clinical term, but what it describes is extraordinary. Somewhere in Eurasia, long before Neanderthals dominated Ice Age Europe, and long before Homo sapiens emerged in Africa, there lived a population of hominins whose genetic line had already been separate for nearly 1 million years, and they did not vanish without a trace. They mixed. One of the leading candidates for this ghost population is a late form of Homo erectus, a species that left Africa nearly 2 million years ago and spread across Asia. Homo erectus fossils have been found in Indonesia, China, and Georgia. They survived far longer than once believed, possibly overlapping in time with Denisovans. If Denisovans encountered a surviving population of erectus-like hominins in Asia, the genetic evidence suggests they did not simply compete, they interbred. This would mean that Denisovans themselves were hybrids, already carrying the DNA of a far older lineage before ever encountering modern humans. The implications are staggering. Human evolution was not a simple branching tree. It was more like a braided river. Populations split apart, drifted genetically, then reconnected thousands of generations later. Traits were exchanged, genes were absorbed, and this process did not happen just once. Recent modeling studies indicate that multiple waves of interbreeding occurred across Eurasia. Neanderthals mixed with Denisovans. Denisovans mixed with super-archaic populations. Later, modern humans mixed with both Neanderthals and Denisovans. Each time, hybrid individuals were born. Some of them survived. Some reproduced. Some pass down fragments of DNA that still circulate in human populations today. In 2018, researchers analyzing a bone fragment from Denisova Cave made another astonishing discovery. The individual, a teenage girl, turned out to be a direct first-generation hybrid. Her mother, a Neanderthal. Her father, a Denisovan. This was not rare experimentation at the edges of species boundaries. It was common enough that a first-generation hybrid appeared in a tiny sample of remains. If Neanderthals and Denisovans interbred frequently, and Denisovans had already absorbed DNA from a super-archaic species, then hybridization was not an exception. It was the rule. But there is a deeper question beneath the genetics. What did these beings look like? We have partial Denisovan fossils, a finger bone, teeth, fragments of jaw and skull from Tibet and Siberia. The Tibetan jaw discovered on the Baishia Karst Plateau shows massive molars and a robust structure unlike modern humans. Yet genetic analysis links it to Denisovans. If Denisovans were already hybrids, carrying DNA from an older lineage, their physical traits may have reflected that complexity. Thicker skulls, larger jaws, distinct facial structures, perhaps a blend of archaic and emerging human features. And somewhere in that mixture, there may have been individuals who were neither fully erectus nor fully Denisovan, not fully anything we can name. The first human who wasn't fully human, a hybrid born of two deeply separated evolutionary paths. For most of the 20th century, anthropologists debated whether different human species could even produce fertile offspring. The biological species concept suggested they might be reproductively isolated. Ancient DNA has overturned that assumption. Not only could they interbreed, they did so repeatedly across vast distances and timescales. Evolution was not a clean succession. It was contact, conflict, exchange, absorption. And this hybrid complexity may explain some of the puzzling gaps in the fossil record. Around 800,000 to 1 million years ago, hominin fossils become sparse in certain regions. Then new forms appear with unusual combinations of traits. Rather than representing sudden evolutionary leaps, these fossils may represent populations shaped by ancient hybridization events. DNA sees what bones alone cannot, and what it sees is a past filled with encounters. But there is still one mystery unresolved. If Denisovans carried super-archaic DNA, and modern humans later absorbed Denisovan DNA, then how much of that million-year-old lineage survives inside us today? To answer that, scientists had to turn to increasingly sensitive genomic tools, methods capable of detecting faint ancestral echoes buried beneath layers of more recent admixture. What they found suggests that the shadow of this ancient hybrid lineage is still present, not as a separate species, but as a fragment woven into our own genetic code. And that discovery changes the way we define what it means to be human. When researchers began isolating Denisovan-derived segments inside modern human genomes, they expected to find scattered neutral fragments, evolutionary leftovers with little consequence. Instead, they found something remarkable. Some of the Denisovan genes carried by modern humans are not random at all. They are functional, powerful, adaptive. In Tibetan populations, a gene known as EPAS, one helps regulate the body's response to low oxygen at high altitudes. Without it, prolonged exposure to thin mountain air can be deadly. Genetic analysis revealed that this version of EPAS1 did not originate in Homo sapiens. It came from Denisovans. And Denisovans, as we now suspect, may have already carried DNA from an even older super-archaic population adapted to harsh Eurasian environments. In other words, when modern humans interbred with Denisovans, we may have inherited survival tools forged through nearly 1 million years of separate evolution, traits shaped by ancient climates, ancient diseases, ancient landscapes. Hybridization was not just an accident, it was an evolutionary advantage. Across Southeast Asia and Oceania, Denisovan DNA influences immune system genes, fat metabolism, and skin biology. Some variants appear to help regulate inflammatory responses. Others may have contributed to adaptation in tropical or high-altitude conditions. These are not trivial inheritances. They are survival strategies. And if Denisovans were themselves hybrids, already carrying genetic material from a lineage that split off close to 1 million years ago, then some of the traits helping modern humans thrive today may trace back to a species we've never formally identified. A species without a name, without a complete skeleton, without a clear place on the evolutionary chart. This challenges a deeply rooted idea in human identity. For centuries, we have searched for the moment humanity became distinct, the spark that separated us from everything that came before. But ancient DNA suggests there was no clean break. There was no single threshold crossed. Instead, humanity emerged through blending, through contact between lineages that had been separate for hundreds of thousands of years. The first human who wasn't fully human may not have been an anomaly. It may have been a common reality. A Denisovan individual carrying super archaic genes, a Neanderthal Denisovan hybrid child, a modern human inheriting DNA from both, each generation slightly different, slightly merged, each carrying fragments of multiple evolutionary histories. The fossil record, once thought to represent isolated species marching in sequence, now looks more like overlapping waves. Some waves collided, some merged, some were absorbed entirely. The ghost population detected in Denisovan genomes has never been definitively matched to a specific fossil. It may represent late surviving Asian Homo erectus. It may represent an unknown branch of early Homo. It may even represent multiple populations. What matters is this: they did not vanish without impact. Their genes moved forward, layer by layer, hybrid by hybrid, until they reached us. This realization reframes extinction itself. Species do not always disappear completely. Sometimes they dissolve. They leave fragments behind, preserved not in stone but in DNA. The boundaries between species blur, and the concept of purity collapses. Modern humans are not the end of a straight line. We are the product of repeated convergence, a genetic mosaic built from ancestors who were not entirely like one another. And this mosaic reaches deeper than we ever imagined, nearly 1 million years deep. As genomic technology advances, scientists are now searching for even older signals, traces of archaic introgression hidden within African populations, where DNA preservation is more challenging and the fossil record more complex. Some preliminary models hint that additional ghost lineages may have contributed to early Homo sapiens within Africa itself. If confirmed, it would mean hybridization was not confined to Eurasia. It was a global pattern, a defining feature of human evolution. The question then becomes not whether the first human was fully human, but whether any human ever was. Because the deeper we look into our genome, the more it resembles a palimpsest, layers of writing scraped away and rewritten over hundreds of thousands of years. Beneath the surface of every cell lies a story of encounter, of survival through mixing, of lineages that should have remained separate, yet didn't. And somewhere in that deep past, long before recorded history, before language as we know it, before art and agriculture and cities, a hybrid child was born, carrying within them two evolutionary worlds, not fully one, not fully the other, but something new, something that would ripple forward through time, through Denisovans, through Neanderthals, through Homo sapiens, until it reached us. The first human who wasn't fully human may never have left behind a complete skeleton for us to name, but their legacy survives in the only place evolution never truly forgets, our DNA. And as science continues to decode that ancient script, one truth becomes unavoidable: humanity was never singular. It was shared,